It's a great pleasure to introduce Dr. Anders Hougaard. He focuses on the use of neuroimaging methods to understand migraine, pathophysiology of migraine, and the triggers for headache. So in some ways, not very festive as a research topic, but Anders completed his medical training and his PhD at the University of Copenhagen, and he's currently an associate professor there. And he's also a consultant neurologist and the head of research at the university's hospital. And his talk is called A Christmas Spirit Network in the Brain. And that is very festive. So please, round of applause, please welcome.
Thank you very much for that kind introduction. I'm not sure if I dare using this clicker here, but let's see. All right. So the reason why I'm here today actually is because some years ago, my colleagues and I got this study published in the British Medical Journal. And this was a study where we managed to locate the Christmas spirit in the human brain. This study received enormous attention from the media, and frankly, this work has been haunting me ever since. So, you know, I'm a neurologist and clinical neuroscientist, and when I go to congresses to present my decent work in headache and neurovascular disorders, people are still like, "Oh, you're that guy with the Christmas study, aren't you?" So while this study really has been obstructing my career, just kidding, I'm very happy that it's brought me here to this wonderful event. And today I even get to tell you the true story behind this study. Right, so let me tell you what we did here. The background, of course, is that the Christmas spirit is a widely known human emotion that is pleasant and warm, and it even arises in a time of the year that's otherwise dark and cold, at least in this part of the world. And frustratingly for us neuroscientists, the neurobiology of this human emotion is really poorly understood. So this motivated us to find a technique to explore this. Fortunately, we have an excellent method called functional MRI that can be used to detect and localize human cognitive function and emotions in the brain. We've seen some examples of that already today. So here we pose the question, where is the Christmas spirit located in the brain? Here you see our experimental setup. We have an MRI scanner, the subject lying here. This is me actually. And you know that this scanner can produce images of the brain, and we can even use it to measure the brain activity here. You notice that the subject is looking into these video goggles that we can use to present visual stimuli. And if we present this rather strong but unspecific visual stimulus, you will see an activation of the visual areas of the brain, naturally. And the underlying physiology of this is that in the resting brain, you have blood flowing through the tissue, and you have hemoglobin that may be bound to oxygen or not. And this deoxygenated hemoglobin is slightly magnetic. This causes a disturbance in the magnetic field of the scanner, and thereby we get less signal from the brain. But when an area of the brain is activated, that area will receive freshly oxygenated blood, and this disturbance caused by the deoxygenated hemoglobin becomes less apparent, and we can detect a higher signal from that area of the brain. So for this study, of course, we needed a more specific kind of visual stimulation. So we selected some images with a strong Christmas content, like this you see here, this cozy, nicely decorated street. This image down here with these delightful traditional Danish Christmas cookies. These were presented to subjects inside the scanner. And of course, we also needed some control images that were virtually identical, but devoid of this Christmas theme. So we had this rather boring street up here and this more everyday like baked goods that you see here. So the subjects were lying inside the scanner, and they were then exposed to these series of images, first Christmas images in a block of around 12 seconds, I think, and then it would switch to these match control images and so on for around 10 minutes. And we would then use statistical tests to look for activation in the brain where the activity was high during the Christmas stimulation and less during the control stimulation without the Christmas content. And to make this study even more specific, we included two groups of people. One group had celebrated Christmas their whole lives, and they would consistently experience the Christmas spirit during the holiday season. And we also included a control group of people who had never celebrated Christmas. And here you see the activity caused by this stimulation paradigm in each of these two groups. You can sort of tell that there's a difference. And finally, we calculated the difference between groups, and behold, here you have the Christmas spirit in the brain. And as you can see, we couldn't locate a Christmas center. So this is more like a widespread network encompassing areas like we have some frontal activation bilaterally, the somatosensory cortex in both hemispheres, and some left parietal lobule activation. So this was really nice and beautiful. And now finally the mystery of Christmas is solved, and everyone can rejoice and enjoy a nice, neurologically well-characterized Christmas. But alas, this study has several limitations that make it really essentially completely useless. I really like these AI-generated images. It's completely nonsense. Why do they have two Christmas trees? Look at that teddy bear. It's really, really quite disturbing. Okay, so this study, I have to stress that this was a real study done in real people with real data. And the methods that we used were standard, very similar to other functional MRI studies. But if you look closely, you will see that something is not really right here. So let's start with some technical limitations. So what we are looking at is not really those nice reconstructed images that you see up there in the corner. But we're rather looking at these images that we acquire during the scans, which are low resolution. We get a lot of these images. We acquire them every 3 seconds, so that's our time resolution, which is also not very optimal to study brain activity. And the signal that we get is really noisy, actually. So of course it's influenced by thoughts and emotions, but much more so by blood pressure, heart rate, movement inside the scanner, and all sorts of things. And what we're really looking at when we're looking at these images are numbers inside these small cubes, 3-dimensional pixels or voxels, we call them. And in such a standard brain image here, we have roughly 150,000 voxels. So if we do this experiments and acquire 200 images during such a scan session, we get roughly 30 million numbers per subject during a scan. And if you know a little bit about statistics, is that when you have such a huge amount of numbers, if you start doing statistics, you have a risk of ending up with false positives. This was a very famous functional MRI study with a single subject. That subject was a frozen Atlantic salmon. And the researchers would place the salmon inside the scanner and subject it to a psychological test. So they would show the salmon images of people in various social situations. And they would ask the salmon to describe what emotions that these people may be experiencing. And as you can see, seemingly there's a very nice brain activation going on associated with this psychological test. And this is a really strong visual effect. So when you're looking at this, you're wondering, is this fish really dead? Or I can see this with my own eyes. Maybe it's really alive. It's like the Monty Python sketch with the dead parrot, you know. So maybe it's just pining for the Norwegian fjords. But really, this salmon is deceased. And this is an ex-salmon. And thus it has no brain activity whatsoever. So we have to remember what we're looking at here, these colorful blobs, are numbers. These are the worst kind of numbers. These are the results of badly performed statistics. These are false positives. And obviously today, these statistical methods have improved and they're less likely to produce false positives, but still functional MRI studies come with a high risk of false positives. Next series of issues with this study, I would call basic design related issues. So we make some assumption in this study. Now we're really dissecting this study to be really boring and rip it all apart. So we assume that these Christmas images that we have and the control images are identical, except that one group of images have a Christmas content and the other do not. But if you look closely, there may be some other systematic differences between these images. Maybe the Christmas images have more red and green color in them, or maybe they're a bit darker. We also assume that these two groups of people are completely identical, except for their festive traditions. But all sorts of factors may influence these very noisy functional MRI signals. And maybe we didn't account for some important differences between the groups. And we don't give any baseline characteristics in the paper at all, and people didn't really think critically about that. So we don't even consider their age, gender, etc., even though those factors may influence what we are getting in terms of functional MRI data here. And also during the scans, this is extremely important. What were they feeling, except that they were looking at these images? What about their heart rate? What about their levels of anxiety by being put inside this machine? People in white coats standing around them. How much did they move? Well, you can account for that to some extent. And you should, because it's very important. So together, these statistical, technical limitations, these basic design-related issues are probably the reason why so many functional MRI studies are not reproducible. So as an example, now I get to highlight some of my own recent research here. So this was a paper that my colleagues and I published a few weeks ago, where we did a functional MRI study in migraine patients, a subtype of migraine called migraine with aura, where we know that these patients have some cortical involvement during the migraine attacks. Here, patients were studied outside of attacks. And this is interesting because many studies have been published in this patient group. All of these previous studies are rather small, maybe include around 20 patients, and all of them describe some kind of difference between patients and controls. But none of the studies report the same differences. None of the studies applied the same methods. And all of these results are really difficult to interpret and to make some clinical significance out of. So here for this study that we did, we tried to reproduce some of these previous findings in migraine with aura. So we did a larger scale study, including 160 patients with this condition. This was a population based study. So these patients were recruited from the general population, and we had a wide range of health related factors that we knew about these patients, so we could account for, of course, their age and gender, but also whether they had hypertension or diabetes, or what kind of medication, the socioeconomic status. So we would carefully match patients and controls for this study. And we could even compare groups of twins, 30 twins, where one group of twins had the condition, migraine with aura, and their co-twins did not. And in this study, we did state-of-the-art analysis. We tried to do all the stuff that was done before, but we found absolutely nothing. So this image is just to show you in the top panel, the patients, and in the lower panel, the controls. And the point here is you cannot tell the difference here. So I'm not saying that all of the previous studies were wrong, but their findings at least were not very robust. And this seems to be the general picture when we look at the functional imaging landscape here. So we have really a reproducibility crisis in functional neuroimaging. Going back to the Christmas study, there are even more issues here because this was a study about emotions. And here I would highlight what I would call some complex design related issues. So we have to ask ourselves, what did these people really experience while they were inside this scanner? Did they feel the Christmas spirit? And did they feel the Christmas spirit for those 12 seconds while they were watching Christmas themed images? And then for the next 12 seconds, they would just feel neutral. And then for the next 12 seconds, they would be feeling the Christmas spirit genuinely. We don't know because we didn't bother to ask them. Could these people have felt something else than the Christmas spirit? The experiment took place during the summer, so maybe they just thought that these images were funny or silly. Maybe they were feeling joy, but without a specific Christmas theme to it. We don't know. So, but we are making the assumption that these people were genuinely feeling the Christmas spirit during this experiment. Then there's an issue I would call brain arithmetic. And here what we do is that we add and subtract these rather complex brain activity patterns. And it's not really clear that this is valid. It's not clear that the mathematical assumptions that would be underlying this are fulfilled. So it's like if you think of something and that causes a brain activity pattern, and then you think of that something more strongly and that causes a different brain activity pattern, then if you add together these activity patterns, would that correspond to you thinking of that thing very strongly? Maybe, maybe not. At least that's controversial, I can say. Then we have some even higher level issues. There are some psychological and philosophical concepts at stake here that are often exploited by the mass media. One of them is neurorealism, and that is the concept that a subjective experience can only be real if we can see it in the brain. So this was a common headline when this study was promoted in the media, that now we know that the Christmas spirit is real, finally, because we can see it in the brain. And that's really a common theme in the media, like this headline you see here, fat really does bring pleasure. So this was from a functional MRI study where people were being given these nice high-fat treats while lying inside the scanner, and you could see their pleasure centers lighting up. So now you finally knew that fat does bring pleasure. You didn't know before. So this is a bit problematic, even especially when this realism, this proof, is just an average of noisy signals causing these colorful blobs on brains. So that proof is maybe not extremely accurate. Next theme is this neuroessentialism, meaning that the essence of a person with that person's thoughts and feelings can be reduced to that person's brain. And philosophically speaking, that may be accurate. But when we're talking about reducing a person into, again, these rather noisy signals that we average, that may not be fair. So when you see this in the media, you can see that the grammatical subject is changed from the person to the brain. So it's no longer this person feels so-and-so during Christmas, but now it's the brain does so-and-so when it's Christmas. And this reduction of the essence of people into blobs on the brain bears some resemblance to the practice of phrenology, which was very popular during especially the 19th century, where personality traits, behavioral traits of people could be determined by the small bumps on their skull in various places. And now the nowadays practice of reducing people into these colorful blobs in various places of their brain has been termed neophrenology or blobology even. So finally, there is the concept of the seductive allure of neuroimaging. And remember the salmon. This is really a strong image. You get the feeling that I can see this with my own eyes. It's true. So these neuroimages with colors on them are really convincing. And there are even psychological studies showing that if a claim is presented, even a controversial claim, if that is accompanied by a neuroimaging picture with these colors on it, it's much more likely to be perceived as true. So this effect has also been termed neuro-rubbish. And I think I was the first person ever to use the term neurobollocks in the British Medical Journal. In conclusion, I don't want to seem like an overly critical Grinch here, because I do strongly believe that functional neuroimaging is a wonderful method and a useful approach to studying the human brain. But we have to remember to be critical here, and we have to know the limitations of these techniques. We have to design studies very carefully to get the proper results. And we have to avoid being seduced by these colorful blobs on brains. So, does this study locate the Christmas spirit in the brain? I would say absolutely not. Does the study further our understanding of what the Christmas spirit really is? Definitely not. Should functional neuroimaging studies be interpreted with care? Yes, indeed. And with this, I wish you all a very happy festive season. Thank you.
So we have time for some questions.
Front here. Thank you. Great talk. I'm very sympathetic to a lot of what you say, but I am a neuroimager, so I feel I have to come push back to some extent.
Great.
I agree very much with your fourth point about neurochronology, neurology, the dangers that those have. Your second and third points about complex design issues, you call them, I think are true of any experimental psychological research on humans.
Right.
So I'm originally a psychologist. All those issues apply to anything, nothing with imaging per se. Trying to understand complex human thoughts is difficult. The first point is the most important one. No doubt the field is facing massive previously problems, and there's a lot of poorly designed, poorly analysed, underpowered studies. So I absolutely agree that's the most important thing, statistics, in the first issue. So to stop us throwing the baby with the bathwater, we need to solve that. I think the main problem is the degrees of freedom analysis. In some ways you could analyse your data. Of course, registering your analysis in advance is a good way to stop that. Now, I would argue that that's true of many fields of neuroscience. Many complex analyses, if you give 2 degrees of freedom, you can find a result if you look hard enough. So just point out it's not unique to neuroimaging.
Yeah.
But otherwise, I agree with you completely.
I absolutely agree, of course. And, really, there's a reproducibility crisis in all of science. We also know in psychology, definitely. And I think that pre-registration of study protocol is just essential for everything, also in neuroimaging.
Hello, hi. I really enjoyed your talk. I really enjoyed it. I think it goes, the points you raised go far beyond the Christmas spirit and neuroimaging. I mean, there are a lot of techniques like proteomics or transcriptomics that encounter the same problems. So my question is, have you used correction for multiple comparisons? And the other half of my question is, do you really believe it? Because to be completely honest, I don't believe it.
So in our Christmas study, yes, we did apply correction for multiple comparisons according to the standard techniques at that time. Now it's 8 years ago. I would say that there's still a high risk probably of obtaining false positives. And if I believe it, no, absolutely not. I believe it's complete rubbish.
If you believe, let's say, Benjamin Holzberg, or if you believe this approach.
Yeah, so if I believe that it's possible to correct appropriately for multiple comparisons in functional neuroimaging studies, yes. Today I use some permutation-based studies that are probably better, but you're never absolutely sure that you're not doing something wrong or insufficiently. But methods are improving, but you have to be critical at all times.
Thanks for the great talk. I found that really interesting. I'm also a neuroimager, but I'm going to be a little bit more optimistic. So instead of this possibly being the Christmas spirit brain, do you think this could just be the holiday brain? Do you reckon there's an Eid spirit? There's a Chinese New Year's spirit in the brain as well?
Yeah, it's absolutely possible. We speculate a lot on that in the paper. I recommend that you read it.
A problem with fMRI that you're looking at a brain that's processed information in milliseconds, 2,000 milliseconds after it. Another of my problems is emotion. I love the experiment. I'll put you in a brain scan, I'll tell you I love you. I need to do it 20 times before I get a signal, by which time I'm so pissed off. I'm looking at a brain that's basically, it's a real serious problem with a monomodal approach to brain imaging, in my humble opinion.
Yeah. Exactly. I completely agree. That's one of my points here. 3 seconds, that's a huge amount of time when you're talking neurosignaling. And these voxels, each of them would contain 600,000 neurons. So we're not being accurate in any way. It's really rough.
One more question.
Do you think that if you would have included a multimodal design, so another neuroimaging technique, and also peripheral markers, so for example, looking at cortisol levels, would your data be more robust or not?
Yes, I think so. It would be good to target this problem from different angles, definitely.
Okay, so with that, massive round of applause. Thank you very much.